We are doing Inside the Lab with the CMBC, and today we're having a short conversation about research with Danny Dilks, Associate Professor in Psychology. So, Danny, just tell us a little bit about yourself.
Sure. I have been at Emory for about 7 years. I came in 2013. Prior to being in the Department of Psychology, I was at MIT as a postdoc and a research scientist with Nancy Kamlisher. I was there for about 5 years, 6 years. And prior to that, I did my PhD at Johns Hopkins with Mike McCloskey and Barbara Landau. I am a cognitive neuroscientist, and my primary method that I used in the lab is Fmri for the most part.
Right. And so, what, if you were going to describe your research generally, what are the major themes of your research? So, what are you really interested in
Yeah,
figuring
good.
out?
So I'm generally interested in three questions. And the first question, or they're all actually related to one bigger question, and that is how it is that we are able to perceive, recognize, understand people, places, and things. The three things, if you will, that we visually encounter that are necessary for our survival. Again, people, faces, places or scenes, and objects. And the first way that I kind of look at that grand question is to look at the brain and ask how the brain is organized with respect to these three domains. And so this addresses an age-old philosophical question of the structure of knowledge. So one idea is that these three things that are crucial for our survival, if indeed are crucial for our survival, might have their own dedicated neural real estate, right? Or even real estate in mind, such that they're separate things, specialized. Jerry Voder called them modular at some point in time. We try not to use that term modular because it's a bit controversial,
Yes.
yeah? So
Uh,
we'll talk about it as specialized. So that's one hypothesis. By looking at cortex, I can look at the mind, the structure of the mind, or the structure of knowledge. And one is that each of these three things, people, places, and things, might have their own private real estate. The other alternative to that is that we don't have these specialized systems, but essentially the brain or mind is a little more equipotent,
right.
where the same machinery is involved in all three of these functions. My research, and I do that first question, looking at the functional organization of the cortex, asking whether these have three specialized systems in the mind, and again, with the method of fMRI, which I talked about. And that's all done in typical adults first. The second question then is another philosophical question related to knowledge, and that is, where does the knowledge come from? So first, what's the structure of knowledge in typical adults, and how do we get there? And this harkens back to, again, two theoretical camps. The first is that of the nativist camp, in that we come to the world with these particular specialties, functions, domains. The other is that it takes extensive experience. And in that case, what I do is I look at typically developing children, mostly, at various ages, and ask, well, do they have this face system and place system and object system, and how does that compare to adults? And then finally, the third question I ask is, once we're all wired up, then how is it, or is it possible then that the brain can change? And that's probably a straw man question, because yes, the brain has to change. There is learning, there's even recovery from stroke. The brain's changing right now in every single one of us, right? It's not changing our percept, but nonetheless, it's changing, right? So to what extent and under what conditions, and what systems can change and what systems can't change, is what I investigate. So again, how the brain is functionally organized, what are the origins of that functional organization, and can that functional organization change once we're all wired up, for example, as a result of neurological insult, for example, stroke. Clear?
Right. Yes. And so the study I think we're going to be talking about today addresses a couple of these questions, right? But mostly the origins
Mhm.
of this
Mhm.
knowledge. So could you tell me that we're referring to this PNAS paper where you studied newborns, right?
Mhm. Mhm.
So if you could tell us who your collaborators were first off, and sort of what was the main question and how you went about, examining that
Yep,
question.
perfect. So first, let me acknowledge the first and second author. The first author is Freddie Camps. Freddie was a graduate student of mine here. He just graduated recently and has taken a postdoc with Rebecca Sachs and Nancy Kantwisher at MIT. The second author is Cassie Hendricks, who is a current grad student here at Emory in the psychology department with her advisor, Patty Brennan, who is the third author on that paper. Cassie is getting ready to defend, and she is taking a postdoc in NYU. So both of those are success stories, and it's beautiful. Without those two, this paper would not have happened. So I have to first say that, of course. And then Patty is on there as well, who gave some good input on it too. So those are the collaborators. Why did we do this? We did this first, I think, because
Yeah, why did you do this?
exactly. Which
Yeah,
we should all ask, actually. We should all ask that about our own stuff.
yeah, like what in the world? Yeah,
Exactly. And probably before the paper even comes out, right? Like, so what? Why are we doing
yeah, like
it? Okay.
why do we care?
So the real motivation to this paper was if we, I had written a review some years ago, and I was looking at the behavioral signatures of face processing in adulthood. For example, we are much worse at recognizing faces upside down than we are upright. This is called the face inversion effect. That happens on faces, and it does not happen on other stuff, or at least to the extent that we see it on faces. For example, if I turn the Eiffel Tower upside down, a picture of the Eiffel Tower upside down and upright, you're going to be equally equally accurate at identifying the upside down one as the inverted one, as the upright one. But that does not happen in faces. You are much worse, yeah? There's also other key signatures of face processing that we can see behaviorally in adults. And we went back in this review and looked at all of these key signatures that people have looked at in kids 5, 4, 3, even some in infancy. And lots of these so-called qualitative signatures in adults, we see really early in development, or early if you're considering 3, 4, 5 early. Yeah? Okay. By contrast, during when I was putting out that review, there had been an imaging paper that had come out saying that the regions, or at least one of these regions, the fusiform face area, or the FFA, that's implicated in lots of these key signatures, is not fully developed until around 10 years old. So I was always puzzled by why isn't the behavior matching the neural?
Right.
That has to happen, yeah? Unless we want to say that maybe the kids are looking behaviorally like the adults, but they're using different machinery to get there. That's certainly possible, yeah? And that's kind of fun. But there were other reasons that I started coming up with, aside from that fun reason, more boring reasons, like is it even the size of the head coil being used, right? In the sense that a small head in a big coil is going to give you a very different answer than an adult head in an adult coil that fits and so on, right? So there are these technical problems, perhaps, why it looks like the neural system that's responsible for the behavioral things doesn't look like adult, yet the behavioral stuff is still there, yeah? So that's always been the question. And as a consequence, then, I thought that I'm going to believe, if you will, the behavioral data over the neural data. And by the way, I told you my primary method is imaging, but imaging It's fickle, and you have to do a lot, uh, like cross-validate and within subject replication and so on to truly believe those
Right.
data. There, you are looking at the brain, and there are hundreds of thousands of voxels that you're looking at, the pixels within the brain that you're
Yeah.
doing analyses on. So you are going to get false positives, 100%. Yeah,
Right.
and
Right.
unless you take great pains to make sure that you're not seeing false positives, which a lot of people don't, unfortunately, then those false positives are out there. And
Right.
so there's my first thing with the seemingly conflicting data between behavioral and imaging was something was wrong with imaging. I'm going to believe the behavior. And
Right.
by the way, we should all believe the behavior in any.
Right.
Okay.
Right. Right.
So in that case, I said, let's see if we can see anything face related really early, not even 3, 4, or 5, but infancy. And while I would have loved, Lynn, to scan infants at 6 days old while they were actually looking at things,
Yeah.
that's even more challenging. We're trying to do that now, not
Yeah.
successfully, but we're trying to work on it. So stay tuned on that. But at least the method that we used here called resting state fMRI, we were able to do because you're able to get that signal or those data from sleeping individuals. So a baby at 6 days old, swaddled, they love to be swaddled, will go fast asleep, and I can collect those data. And now I can really go as early as I'd love.
So you're the infants that you put into the the scanner were 6 days. How old were
Uh, the,
they?
yes, well, the earliest was 6
6
days,
days. Okay.
and the oldest was 57 days. So fair enough,
Okay.
that's kind of a big range, but in this
Right.
case, you you take the infants you can. Yeah.
Yeah. Yeah. Yeah.
And you can always look at the really young versus the older and so on post hoc. So
Right. Right. So you put them in the scanner, they're swaddled up, they're sleeping, and you're looking at resting state. And so
sure.
what did you guys find?
Yep, good. So the hypothesis, uh, let's go back to that first question I told you, the
Yeah.
lab process. In adult, there are at least 2 regions
Right.
in the adult brain, cortical regions, that are selected for faces. That means in every one of you, everybody that's listening to
Yeah.
this, if I put you in the scanner and I show you images of faces and images of objects, and I contrast those two and ask which part of the cortex is
Mm-hmm.
more activated, if you will, yeah,
Mm-hmm.
when you're looking at faces than objects, you're going to get at least these 2 regions, one called diffusiform face area, FFA, and another one called the occipital face area, OFA. Okay.
Right.
Aside from that, in you as well, if in that same scan to find these regions, I now put pictures of places or scenes in addition to the faces and objects. And I now look at parts of the brain that are more activated when you're looking at pictures of places like Eiffel Tower versus the objects. I'm going to get
Mm-hmm.
a different network, a different system than the
Right.
face
Right.
system. And those 2 of those regions within this larger network are called the parahippocampal place area, the PPA, and the retrosplenial complex are the RSC. So we know in adults, Lynn, you,
Yeah.
me, all of us, we've got these 2 regions that are face selective and these 2 regions that are scene selective.
Mm-hmm.
And we also know that those 2, the 2 face regions are connected to each other, and the
Right.
scene regions are connected to each other, and they're not connected across each other. Yeah.
Got it.
So we know that's how it is in adult. So we used resting state asking that same question in baby. Will we indeed see that what's going to ultimately become or already is in baby, right? We don't know that because I can't keep them awake looking at pictures of
Right.
places.
Right. Right.
But is that connection there at least between the face, what's going to become the face system,
Right. Right.
and is the connection between the scene system, PPA and RC there, and the cross connection not there, right? As early as 6 days, essentially.
Right.
On
Right.
average, 27 days.
Right. So
And
little.
so little,
Yeah.
totally
Yeah.
little,
Yeah.
totally little. So the kids, like you said, were swaddled. They were put in the scanner. As long as that infant stayed asleep, I scanned them. So if that baby stayed asleep for 1.5 hours, we got 1.5 hours worth of data, which
Okay.
double, 3 times, 4 times what some other people have done when they
Right.
were
Right.
baby. Okay, so that's number one. So, and the reason why I did that, Lynn, is because I also wanted to be able to, in at least some of those infants where we got a whole lot of data, I wanted to be able to split those data within
Right.
that child and show that same exact effect in half of the data, in one half of the data and the other
Right.
half of the data. So I have a within subject replication. This is one way, by the way, that we can make sure that it's not false positives that I was talking
That's right. That's right. So you're predicting your own, you're using one set of data to predict another set of data.
100%.
Yeah.
It's a
Right.
very widely known thing in neuroscience with animal stuff, yeah, and now becoming much more popular, this cross-validation technique in human
Yeah.
neurology.
Yeah.
Thank God. Okay. So what did we find, Lynn? We in fact found that in babies average 27 days old, yeah.
Yeah.
That yes, indeed, that face system is already connected like an adult, and the scene system is already connected, and they are not connected to each other. So it's as if the baby's coming to the world, or even with limited experience, that is already pre-wired. So imagine it's kind of the highways already there. The towns aren't built on each of the highways, or they're partially built or whatever, but that highway is there, meaning that this town is going to be connected to this town, and this town is going to be connected to another town because there's the highway, and there's no highway that goes across those two neighborhoods.
So the idea would be that the infants come prepared
100%.
to see faces and to see scenes.
Yeah.
And they have neural architecture that may be devoted to those.
That's right.
Those types of stimuli, those very biologically and relevant stimuli.
That's right. And I'd be even a little stronger, and I would not say maybe.
Yeah,
of course.
Yeah, right. It is, right, yeah.
They are pre-wired for these two to become a particular thing, and these two to become another thing. And that leads to the next fun question, I think, of what you're alluding to, is that, well, why is it, right? So because these two are connected, it's guaranteeing that they're going to become the same thing.
Right.
And these two are connected to each other and not connected to these, that these two are going to become the same thing.
Yeah.
Now, the fun, cool question, though, is why is it that whatever these two are ensured to become, why is it that become always face? Yeah. And that these two that are ensured to become another thing, because they're not connected with these, why do they always become seen, right? And why am I saying, by the way, always become face and always become seen? Because that's how it is in us, in adults.
Right.
Never, ever, in any person I've ever scanned, is the face system in a completely wildly different part of the cortex in any of us, yeah?
Right.
Or never is the face system a place system in another person. It just doesn't happen, yeah?
So then is the question, what is it about scenes and this particular part of these cortical regions, and what is it about faces and these part that makes these parts best able to compute faces and these parts best able?
Exactly.
Is that the idea?
That's totally the idea. And so we try to address that, yeah? So if you read the paper, you'll end up seeing us calling it like the proto-FFA and the proto-OFA. And the reason why we did that is because, again, I can't keep these babies that young awake to see that that's actually the FFA. And by the way, it might not even be actually the FFA yet. Yeah.
Right.
So that might be prior experience. I don't know, right? I've scanned 4 months old babies and actually kept them awake. And they do have FFA and OFA and PPA and RSC, but it's not as selective as ours. It's not as cranked up as ours, yeah? So in that sense, I was calling it proto because we know at 4 months old, it's still not fully adult-like, so it's not going to be adult-like at 27 days old, yeah? That's number one. And number two, it might not even be there, right? But I know that what I'm calling the proto-OFA and FFA the babies are going to become that for sure, because I dropped down. So there's there was another person, Josh Julian, who'd been an undergrad or a master's student with me while I was at MIT. Yeah. He asked, in adults, for example, we all have an FFA and he was asking how consistent, really anatomically, right? We'll find it in everybody, nearly everybody, right? But how consistent it is anatomically. So he made an overlap map and just put a parcel around where 95% of the peoples or 98% of the people FFA fall. And so that's what I dropped down with some transformation onto baby brain. So I know with at least 95% certainty that that's region's going to be FFA.
Right.
Okay, good. But now the question is, what you're asking, Lynn, is gorgeous, right? It's that, why is this one always become face?
Yeah.
And because it's not full-blown face yet, right?
Right.
And so one idea, we just took a step back from the data, and we're not the only people that hypothesized this. Alfonso Karamazov, Marge Livingstone, Brad Mahon, they've hypothesized why these become what they've done in the following way as well. So if we just, all of us, you, the audience that's listening to this, in our daily lives, we, when we look at a face, we fixate the face. We look at the face. We foveate the face, yeah? By contrast, places, scenes, are always in the periphery. Right? You don't fixate a place. Like if I told you, fixate the room, right? You can't, because it's beyond your field of vision. Yeah?
That's right. You're scanning all over the place, right? Yeah.
So intuitively, you're always looking at a face, fixating a face, while the scene is always in peripheral vision. Now, in early vision, by the way, early visual cortex, like called V1.
Yeah.
Primary visual cortex. We've got bits of primary visual cortex that are selective to what you're looking at, fixating, and another part of V1 that's selective for what's in the periphery. Yeah?
Yeah.
We hypothesized, along with others, like Marge and Alfonso and so on, that maybe face always becomes face because it's preferentially hooked up to foveal V1.
Nice. That's nice.
And the consequence of always looking at the face, that information's already thrown on that highway and it only can go one way. And you only look at faces, so that's becoming face. And those two are going to become face because you're born with them, or it takes very little experience for them to be already pre-wired. By contrast, we hypothesize that what's going to become the scene network, is preferentially hooked up to the peripheral.
Right.
Yeah? And that's indeed what we found.
Ah, very nice.
Right? So there's a little bit of a problem, and you're probably going to pick that up, So I'm going to cut to the chase before you even get me. You are probably thinking that, well, you buy that peripheral, right, that scenes are always in the periphery, and that we fixate faces. We also fixate objects, right? When I ask you to get the cup, look at the cup, you look at the cup. And so on. So then why is the face network become face? Because you also fixate objects. So then in us, if that's true, if that's the only driver that makes that face, that just can't be true, right? Or if it were true, the face system wouldn't be a face system, it would be a face and object system, and that's not true in adulthood. So it's not enough to say that it's just that bottom-up input that is making the functions proper. You have to posit something else, at least for place. For place, you don't have to posit anything else, except the bottom-up. But for
so what do you think that other thing is?
Yeah, exactly. So there are there's some recent evidence from Rebecca Sachs' lab, where Freddie Camps, the first is now, and her hypothesis that there's a bit of frontal cortex that is particularly involved early, that is early in childhood, infancy, probably, that is involved in the social interaction between mother and child. So imagine you've got now what the baby's fixating on, face, combined with information, and that frontal bit is connected to that face network. So imagine the confluence or the combination of those two inputs. I'm having social interaction with this thing, plus I'm looking at it, that makes face. And now object, you might have only the fovea, but there's no social interaction with an object, So my guess at least one hypothesis, and there could be many, right, is that there is going to be that second input, and that second input, at least one of the hypotheses that makes big sense to me is that that social interaction bit could be that second source that actually makes face space.
Right, right. So there's some kind of social salience or
Yep.
social significance,
Totally.
which makes sense, right?
Totally.
So if I were a critic,
yeah, please.
right? So, you know, you've got this. So this was published in the Proceedings of the National, right?
Yeah.
Academy, PNAS of
Yeah.
Sciences,
Yeah.
right? Did I already say that? I hope.
Yeah.
And so what, you've gotten a lot of attention for this study because it's really, you're and your colleagues. So what would be the pushback? Is it that there's lots of experience early on even? Is that's the
I was a bit careful in writing it, but not always that careful on purpose, right? I always try to take a little bit of a stronger position because that's kind of fun. And with my general science, I think if you take a little slightly extreme position, right, then it gives something for people to go after, and that's science, right? We should be
we're generating conversations that way.
Totally.
Right. Discussions.
And I think that's healthy science. Yeah.
Yeah.
But so I push it where I really end up, I think, even in the abstract saying, and probably innate. Yeah.
Yeah. Yeah.
Not possibly, although the editors may have maybe changed it. I don't know.
Yeah.
Not possibly, but probably innate, right?
Right.
And can I say that? No, come on, I can't say that, right?
Right.
For real, right? Because it could be 6 days of experience, Lynn, right? That is required for this to become pre-wired. I can never rule that out, right? In these, by contrast, I could rule it out if I did a controlled rearing experiment. So let's do a thought experiment for a second. Let's say that we raised a child, which we cannot, right?
And we're not advocating.
We're not advocating
There's no advocating here.
We're not advocating that, right? But theoretically, if we could a child having never seen a face from birth, but importantly seeing everything else like objects and scenes, right? That's a proper controlled rearing experiment. Then we scan that child having never seen a face, and if we still see what's going to become OFA, FFA, and OFA, the face network is there, having never seen a face, then we're getting experience out altogether, right? That's how we really, really address this. Because we can't do that, what researchers typically do is we just go as young as we can possibly go.
That's right. Yeah.
But interestingly, Lynn, and I think you know this already, but maybe the audience members don't, there are some researchers who have done these controlled rearing experiments in macaque monkey.
Right.
And Marge Livingstone was one of those recently that did this, where she raised some monkeys. Sugita was another one some years ago in Japan that did this. But she raised monkeys having never been exposed to faces. And then she asked about these face regions. She did not ask about the connectivity. She was asking about the regions, if I remember properly, And what she had found is that without experience, these monkeys do not have the fusiform face area, for example, or what would maybe be the homologue of the fusiform.
Right.
And so that's really how I'd have to do this, right? I would love to maybe hook up with Marge or some colleagues here, right? Jocelyn and I had talked about this. Jocelyn Bachevalier talked about this years ago, trying to raise monkeys with no experience with faces. It's logistically a nightmare, but that's not a reason not to do it. But I would love to see in monkeys who have never been exposed to faces, whether that connection is still there. If that's true, yes, I am saying that it's innate.
There.
There.
So, I know I've kept you a long time already, and I'm sure you have other things to do.
More Zoom meetings.
More Zoom meetings.
What else?
So what is experience doing then?
Yeah, good.
Right.
So if one reads this paper, you will see that while the connections are there, they are present in infants. When we compared them and they look just like the adult qualitatively. And that these two are connected to each other, these two are connected to each other, and you don't have the cross connections. There's still some development. Those connections in the infants are still not quantitatively
Right.
the same as the
Right.
adult.
Right.
So there is
Right.
some development there. There's also got to be development within the regions in and of themselves, right? Because I told
Right.
you already that in the ones where I've done actual fMRI, not resting state fMRI, in the 4-month-olds, these regions are still face selective. Yeah, they're still responding more to faces than objects. But
Yeah.
in you, for example, yours responds about 2 to 4 times more to images of faces than objects. But in a
Right.
4-month-old, it might respond only 1.5 times more. Right? So it's not a
Ah,
full-blown already face system. And the connections, according to this paper at least, right, are also, while still there and qualitatively adult-like,
right.
they're not quantitative. So
Right.
experience still plays a role, right? Please
Of
don't think
course it
that
does,
doesn't,
right? Yeah.
right? And so
Yeah.
what my idea of experience, at least in these systems, by the way, experience is going to have a much larger role in other things
Yeah.
like language systems. It has to, right? Because
Yeah.
language has not been around that long. It
Yeah.
is experience that is driving language and, for example, the visual word formary and so on, right? This
Right. Right.
cannot be innate in the sense of evolutionarily old, right? And we
Right.
come to the
That's
world.
right.
But I think that these regions are there really early. And again, I would say at birth,
Right.
in a proto sense, in a primitive sense. Yeah, it's still going to do its thing. However, it does that. Yeah. And what experience does, at least in these early domain specific functions, is fine tunes it.
Yeah. That's right. That's
Yep.
right. Excellent. Danny, thank you
This was
so
great.
much. This was fun. We should just do this for fun.
Exactly.
Yeah.
I
Yeah.
love this. I love this, and I hope you're
Yeah.
successful and you get to interview a lot more people. It's super fun.
Yeah. Yeah.